Okay, so you're in med school or maybe starting in a clinical lab and you hear cell culture. Ever get that feeling like, whoa, what is this exactly? Staring at those flasks for the first time.
Oh yeah, it can definitely feel a bit intimidating, like this whole hidden world in the lab.
Right. But honestly, it's not some super complex mystery. It's fundamental, a core technique behind so much biology and medicine today.
Absolutely. Think of it like this. You're essentially taking cells, maybe from an animal, maybe a plant, and you're setting up a little controlled environment for them outside the body, a place where they can grow.
A little cellular home away from home.
Exactly. And for this deep dive, we're really going to break down what that involves. We've pulled from some great foundational guides on cell culture basics.
Yeah, the idea is to give you, especially if you're a student just getting started, a really solid, clear overview, a shortcut to understanding the essentials without getting bogged down.
That's the mission. Make it engaging, make it clear. So, cell culture. What are we actually doing?
Let's start there, the absolute basic.
Okay. So, at its heart, it's removing cells from that organism, right? And you can get these cells directly from a piece of tissue.
Like a biopsy sample.
Sort of, yeah. You take tissue, and then you need to break it down, usually with enzymes or maybe some gentle mechanical methods to get individual cells. Or you can work with cells that are already established, cell lines or strains that have been growing in labs, sometimes for years. They're like well-characterized cellular populations.
Okay, so either fresh from the source or from an existing lab stock.
Precisely. And that first culture, the one straight from the tissue, that's called a primary culture.
Primary, got it.
But those cells, they grow, they divide. Eventually, they fill up their container, the flask or dish. We call that point confluence.
Ah, so they run out of space.
Exactly. And that's when you need to do something called subculturing or passaging. It's basically splitting the culture, taking some cells and moving them into a new vessel with fresh food, fresh medium.
Like replanting seedlings so they have room to grow bigger.
That's a great analogy. It gives them space, fresh nutrients. It keeps them in that active growth phase, which is usually what you want for experiments. Keeps things consistent.
Makes sense. Keep them happy and multiplying. Okay, so what do these cells actually need in their little artificial world? It's not just a sterile box, is it?
Oh, definitely not. It's quite specific, actually, and it does vary a bit depending on the cell type, but the core requirements are always there.
Like what?
Well, first they need a suitable container, the culture vessel. Then, crucially, they need a growth medium. Think of it as their liquid food.
Packed with goodies, I assume.
Absolutely. It has to provide all the essentials, amino acids, carbohydrates, vitamins, minerals, all the building blocks.
Okay.
But often that's not enough. They also need signaling molecules, things like growth factors, hormones that tell them what to do, when to divide, and gases.
Gases, like oxygen.
Yep, oxygen, usually carbon dioxide too, in the right balance. And then there's the whole physicochemical environment we have to control. Things like pH, how acidic or alkaline it is, osmotic pressure, the salt balance, and of course temperature. It all has to be carefully regulated, mimicking their natural conditions as closely as possible.
Wow, it really is like setting up a perfectly controlled microenvironment. A cellular spa, maybe?
Yeah, you could say that, a very precisely controlled spa.
No, I've heard terms like adherent and suspension cultures. What's the difference there?
Yeah, that's about how they like to grow. Most cells that come from solid tissues, like skin or liver cells from vertebrates, are anchorage dependent.
Meaning they need to stick to something.
Exactly. They need to attach to a surface the bottom of the flask or dish to spread out and divide. That's adherent or monolayer culture. They grow flat in a single layer.
Okay. And suspension.
Suspension cultures are for cells that are happy floating freely in the liquid medium. Think of blood cells, for instance. They don't naturally stick to surfaces in the body, so they don't need to in culture either.
Different lifestyles, different setups. Makes sense.
Our source also mentioned finite versus continuous cell lines. What's that about?
That's a really important distinction early on. Finite cell lines are basically normal cells taken from tissue. They behave like normal cells in that they can only divide a limited number of times.
They have a built-in expiration date.
Sort of, yeah. They reach a point called senescence where they just stop dividing, they get old. Continuous cell lines, though, are different. They've usually undergone some kind of change, a transformation.
Like becoming cancerous.
Often, yes. Or maybe they've been altered in the lab. The key thing is they've bypassed that normal senescence limit. They can divide indefinitely as long as you give them the right conditions. They're essentially immortalized.
Okay, so finite lines have a limited run. Continuous lines can go on, useful for different things, I imagine.
Definitely. Continuous lines are workhorses for a lot of research because you have potentially unlimited supply. Right, now let's shift to the lab itself. A cell culture lab sounds like it needs to be, well, super clean and controlled. What are the big safety considerations?
Safety is absolutely critical. You've got your standard lab hazards, sure, but with cell culture, you're working with living biological material.
Which adds extra risks.
It does. Things like accidental needle sticks, spills or splashes generating aerosols that you could inhale, handling potentially contaminated materials or sharps. There's a risk of exposure to biological agents.
Yikes. Okay, so what's the main goal of safety protocols here?
The number one goal is biosafety, reducing or eliminating any potential exposure of the lab personnel and also the outside environment to harmful biological agents.
How do you achieve that?
It all comes down to strict adherence to good microbiological practices and, crucially, aseptic technique. We'll talk more about that, but it's fundamental.
I've also seen these biosafety levels mentioned, BSL 1, 2, 3, 4. What do those mean?
Those are standard classifications defined by bodies like the CDC and NIH here in the US. They basically rank the level of risk associated with the biological agents you're working with.
So BSL 1 is low risk, BSL 4 is high risk.
Exactly. BSL 1 is for agents generally not known to cause disease in healthy adults. Think basic teaching labs. BSL 4 is for the really dangerous exotic pathogens, maximum containment required.
And where does most cell culture work fall?
Thankfully, most standard cell culture, especially with well-characterized lines, falls into BSL 1 or BSL 2. BSL 2 requires a bit more precaution, dealing with agents that pose a moderate potential hazard. The level dictates the specific safety practices, equipment, and facility design needed.
Provides a clear safety framework.
Yes, sir.
Now, those special hose biosafety cabinets, what's their role? They seem pretty important.
They are arguably the single most critical piece of safety equipment for cell culture. They do two main things. One, they protect you, the worker, by containing any aerosols or splashes generated inside. Two, they protect your culture by providing a sterile, particle-free work environment.
A two-way shield.
Precisely. Especially the Class 2 cabinets, which are the most common type for BSL 1, 2, and even some BSL 3 work. They maintain that sterile field for your cells while keeping potential hazards contained.
And the source stressed having a dedicated aseptic work area too.
Yes, ideally. Having a separate room just for tissue culture is best. It minimizes foot traffic and reduces the chances of contaminants drifting in from other lab areas. If you can't have a whole room, at least a clearly designated separate area within the main lab is essential for all the sterile handling, incubation, and storage. The biosafety cabinet is the heart of that zone.
Got it. Dedicated space, proper containment. Okay, so we have the safe space. What about the gear? What equipment is essential for a cell culture lab?
Okay, the basic toolkit usually includes that biosafety cabinet we just talked about, an incubator, typically one that's humidified and controls CO2 levels.
For that pH control we mentioned.
Exactly. Then you need a water bath for warming up your media and reagents quickly, a centrifuge for spinning down cells, a refrigerator, and usually a Netaful 20 degrees Celsius freezer for storing supplies.
What else?
You need a way to count cells traditionally, a hemicytometer and a microscope, or maybe an automated cell counter. An inverted microscope is key, so you can look at the cells through the bottom of the flask without opening it.
Ah, clever.
And for long-term storage, a liquid nitrogen freezer for cryopreservation, plus an autoclave for sterilizing equipment and media and for decontaminating biological waste.
That's a fair bit of kit. Any more advanced stuff?
Oh yeah, depending on the work, you might have vacuum aspiration pumps for removing liquids easily, precise pH meters, maybe advanced microscopes like confocal for detailed imaging, or flow cytometers for analyzing individual cells.
The consumables, the everyday stuff.
Right. Your culture vessels, flasks, petri dishes, multi-well plates, pipettes, lots and lots of sterile pipettes, and pipetters for handling liquids accurately, syringes, needles maybe, biohazard waste containers, and of course the media, serum, various reagents, and the cells themselves.
Okay, quite a shopping list. Now, we keep coming back to this term, aseptic technique. You said it's the bedrock. Why is it so critical?
Because your cell cultures are basically a nutrient-rich soup sitting at a nice warm temperature. It's the perfect breeding ground for unwanted guests, bacteria, fungi, yeasts.
Things that are everywhere in the environment.
Everywhere. On your hands, in the air, on surfaces, in non-sterile supplies. If those get into your culture, they can quickly outgrow your cells, ruin your experiment, or produce toxins. Aseptic technique is the set of rules you follow to prevent that from happening.
So it's like defensive driving for cell culture, always anticipating contamination.
That's a good way to put it. It's about creating and maintaining a sterile barrier between the outside world and your culture. It involves several key things.
Okay, break it down for us. What's principle number one?
A sterile work area. That means your biosafety cabinet. You must clean it before and after every single use. Usually wipe it down thoroughly with 70% ethanol.
Every time.
Every single time. And keep the area around it clean too. Inside the hood, keep it uncluttered. Only have what you absolutely need for the task at hand in there, and keep everything visible.
Makes sense. Keep it minimal.
Some hoods have UV lights for sterilization between uses, but that doesn't replace wiping. And interestingly, using an open flame, like a Bunsen burner, inside a modern biosafety cabinet is usually discouraged now. It disrupts the protective airflow pattern.
Oh, really? I thought that was standard.
It used to be more common, but the airflow is key. And always keep the hood running while you're working, and usually for a bit before and after too.
Got it. Clean hood, good airflow. What's next?
Personal hygiene. Sounds basic, but it's vital. Wash your hands thoroughly before you start. Wear gloves, sterile gloves often, and a clean lab coat. This minimizes you shedding microbes from your skin or clothes into the work area.
Protecting the culture from us.
Pretty much. Then sterile supplies. Your media, your pipettes, your flasks, they all need to be sterile to begin with. Commercial stuff is usually guaranteed sterile, but you still have to handle it correctly.
How so?
Wipe down bottles and containers with ethanol before putting them in the hood. Don't leave bottles open. Never pour directly from a stock bottle. If you can help it, always use a sterile pipette.
Yeah.
Use each sterile pipette only once. Don't unwrap them until you're ready to use them.
One pipette, one use. No exceptions.
No exceptions. It's all about minimizing any chance for airborne or contact contamination. Be meticulous. Wipe your gloved hands with ethanol frequently too.
Constant vigilance,
Yeah.
like you said. Okay, so despite best efforts, contamination sometimes still happens. What are we looking for? What are the usual suspects?
Yeah, it's a common problem, unfortunately. The main biological contaminants are things like bacteria, molds, yeast, sometimes viruses, this other tricky one called mycoplasma, and even cross-contamination from other cell lines in the lab.
Okay, let's take those one by one. How do you spot bacteria?
Bacteria are often pretty obvious relatively quickly. Your nice, clear culture medium will suddenly look cloudy or turbid. Sometimes you see a thin film on the surface.
Like a scum.
Sort of, yeah. And the color of the medium might change, usually turning yellowish, because the bacteria produce acids and lower the pH. Under the microscope, you'll see tiny dots or rods, often moving, between your much larger cells.
Pretty clear signs. What about yeasts?
Yeasts can also make the medium turbid. The pH change might be less dramatic initially. Sometimes it even goes up later. Microscopically, yeasts look like small, oval, or round particles, often bigger than bacteria, and you might see them budding little daughter cells pinching off.
Okay, different look under the scope. Molds. The source didn't say much.
Molds are fungi, like yeasts, but they grow differently. You usually see fuzzy patches, often floating on the medium or growing up the sides of the flask. Microscopically, they look like long, branching filaments called hyphae, usually quite visible to the naked eye once they get going.
Hard to miss the fuzzy stuff. Now, viruses. They sound trickier.
Much trickier. They're too small to see with a regular light microscope because they replicate inside your cells. Often, there are no obvious visual signs initially.
So how do you know they're there?
You might see subtle changes in cell health or behavior, or maybe characteristic cytopathic effects if the virus damages the cells. But definitive detection usually needs specialized methods like electron microscopy, antibody staining, ELISA, or PCR tests for viral DNA or RNA.
The hidden enemy. And a risk to the researcher too, potentially.
Yes, especially if you're working with human or primate cells that could carry viruses infectious to humans. That's why sourcing cells carefully and using appropriate biosafety levels are so important.
Makes sense. What about mycoplasma? Another tricky one.
Very tricky. They're tiny bacteria without cell walls, so they don't cause the usual turbidity. But they can really mess with your cells, change growth rates, alter metabolism, affect experimental results, all without obvious signs. You need specific tests, usually PCR-based or special culture methods, to detect them reliably.
Oh, sneaky. And the last one, cross-contamination.
Yeah, this is a huge problem for research integrity. It's when one cell line accidentally gets mixed into another culture. fast-growing lines, like the famous HeLa cells, can easily take over slower-growing cultures without you realizing it initially.
So your experiment might be completely invalid.
Exactly. It undermines the whole study. Prevention is key. Get cells from reputable sources, cell banks. Handle only one cell line at a time in the hood if possible. Be meticulous with labeling and aseptic technique. And periodically check your cell line's identity using methods like DNA fingerprinting or karyotyping.
Wow. Okay, a lot to watch out for on the contamination front. So assuming you've got clean cultures, how do you choose the right cells for your experiment and keep them happy long term?
Choosing the line depends entirely on your research question, of course. What biological system are you trying model? What process are you studying? Suppliers like Invitrogen offer a huge range. Once you have them, keeping them happy is all about maintaining that optimal environment we talked about. And the culture medium is central to that, apart from temperature.
Right, the cell food, you mentioned different types.
Yeah, broadly you have basal media, the basic nutrient mix. Then reduced serum media and serum-free media. Serum, usually fetal bovine serum, FBS, is a really common addition to basic media.
Why add serum?
It's like a super supplement, packed with growth factors, hormones, attachment factors, minerals, lots of things that many cells need to thrive, especially primary cells.
But there are downsides.
Yes, serum is expensive. Its composition can vary a lot from batch to batch, which affects reproducibility and is a potential source of contamination itself, like viruses or mycoplasma. That's why people develop reduced serum or completely serum-free media for more defined, consistent conditions, though getting cells adapted to them can sometimes be tricky.
A trade-off then. Okay, what about the physical side? pH, CO2, temperature.
For most mammalian cells, you're aiming for a physiological pH, around 7.4. This is usually maintained by a buffer system in the medium, most commonly bicarbonate, which needs a controlled atmosphere of CO2, typically 5%, to work properly, hence the CO2 incubator.
Ah, that's why the CO2 is needed.
Exactly. Insect cells, though, often prefer a slightly lower pH, maybe around 6.2, and they usually don't need added CO2. Temperature-wise, you match the host. Mammalian cells, 36-37 degrees C. Insect cells, cooler, around 27 C. And you need to keep it stable. Deviations stress the cells, change their behavior, maybe even kill them. Overheating is usually worse than being slightly too cool.
Got it. Keep it steady, keep it right for the specific cell type.
Yeah.
Now you mentioned checking morphology. What does that mean and why do it?
Morphology is just the cell's shape and appearance. Looking at your cells regularly under the microscope is super important. It's your first line of defense for spotting problems.
What kind of problems?
Early signs of stress or contamination. Are the cells looking grainy? Are the adherent cells starting to detach? Are there little bubbles, vacuoles appearing inside them? These subtle changes can tell you something's wrong, maybe contamination, maybe the medium's depleted, maybe they're getting old before it becomes a total disaster.
Learning the visual language of your cells.
Exactly. The source even showed examples. Healthy 293 cells look like this, adherent or suspension. Healthy SF21 insect cells look like that. SF9 cells attach well and form nice monolayers. Knowing what normal looks like for your cells is key to spotting abnormal quickly.
Right. Okay, so we're keeping them happy, watching their morphology. What happens when they grow too much? They reach that confluence point, that subculturing time.
Yes, exactly. Subculturing or passaging is how you keep the cell line going. Cells grow, fill the available space, or use up the nutrients. Their growth slows down. You need to dilute them, transfer a portion to a new flask with fresh medium to keep them actively proliferating.
Give them a fresh start. When's the best time to do it?
Ideally, you want to passage them when they're still in their active growth phase, the logarithmic or log phase, before they hit full confluence.
Why before?
For adherent cells, especially normal ones, hitting confluence can trigger contact inhibition. They stop dividing because they're touching each other too much. This can make them slow to start growing again after passaging. Even for cells without strong contact inhibition, overcrowding leads to nutrient depletion, waste buildup. It stresses them out.
And for suspension cells?
Similar idea. If the density gets too high, the medium gets exhausted, cells might clump, growth slows. You want to catch them before that happens. For insect cells, the advice can be a bit more nuanced. Sometimes passaging at confluence helps detach sticky cells, but doing it repeatedly at high density isn't good. And passaging too early, when they're too sparse, can also be bad for some cell types.
So there's a sweet spot density-wise.
Definitely. And keeping track is important. Our source mentioned pH can be an indicator too.
How so?
As cells metabolize, they usually produce acidic waste products, so the medium pH tends to drop as the cell density increases. You'll see the color indicator in the medium change, usually towards yellow for mammalian cells. Insect cells might show a slight rise first, then a drop. It's another clue, along with visual density.
So observation and timing are key. A schedule helps.
Absolutely. A regular subculture schedule is crucial for consistency. And keeping a detailed logbook, dates, media lot numbers, split ratios, cell appearance, essential for troubleshooting and reproducibility. Plan your experiments around that schedule. Try not to passage cells when they're just starting out lag phase. We're already overcrowded.
Right. Plan ahead. Okay, we know when, now how. What's the actual process for adherent cells? Getting them off the plastic.
Yeah, for adherent cells, you first need to detach them. Most common way is using enzymes like trypsin or a gentler alternative called TrPLE. These enzymes chew up the proteins that glue the cells to the flask surface.
So you add the enzyme, wait a bit.
Yep. First, you carefully suck off the old medium, then maybe a quick rinse with a special salt solution, one without calcium and magnesium because those ions interfere with trypsin. Then add a small amount of pre-warmed enzyme solution, just enough to cover the cell layer.
And incubate.
Usually, yeah, for a few minutes at 37 degrees C, you watch under the microscope. When most cells round up and detach, maybe give the flask a gentle tap, you stop the process.
How do you stop the enzyme?
Add complete growth medium, the kind with serum in it. Serum contains inhibitors that neutralize the trypsin. Then you collect all that liquid with the floating cells, usually into a centrifuge tube.
And spin them down.
Right. Centrifuge gently to pellet the cells, pour off the supernatant, the liquid containing the old medium and enzyme, then resuspend the cell pellet in a known volume of fresh, warm, complete medium.
Ready for the next step.
Almost. Now you usually need to count them. Take a small sample, mix it with trypan blue dye maybe to check viability.
We'll come back to counting.
Okay, so you count them, figure out the concentration, then you calculate how much of that cell suspension you need to add to your new flasks, along with fresh medium, to get the right starting density, the seeding density. Then you transfer it, label carefully, and back into the incubator they go.
Quite a few steps. Any major differences for adherent insect cells?
Some key ones. Remember, lower temperature, 27 degrees C, no CO2, different media formulations. Detachment can sometimes be trickier, especially in serum-free media, might need shaking or scraping more often, and don't seed them too sparsely. Below about 20% confluence can hinder their growth.
Okay, nuances for the insect crowd. What about suspension cells? That sounds easier.
It generally is, yeah. No detachment step needed. For cells in shaker flasks, you just take a sample for counting, calculate how much of the old culture to transfer to a new flask, and how much fresh, warm medium to add to reach your target volume and seeding density.
Simpler. Any tips for shaker flasks?
Use flasks without baffles those ridges inside some flasks. They create too much turbulence and shear stress, which can damage the cells. Make sure the cap is slightly loose for gas exchange.
And spinner flasks, those have moving parts, right?
Right, they have a magnetic impeller that gently stirs the medium, keeping cells suspended and improving aeration, allows for higher densities. The passaging process is similar: sample, count, calculate, dilute into a new flask with fresh medium. Just need to make sure the spinner is working right and the medium volume is correct for the flask size to allow oxygen transfer. There are recommended minimum volumes.
Makes sense. And suspension insect cells. Anything special?
Again, 27 degrees C, no CO2, insect-specific media. You often don't need a full medium change when passaging suspension insect cells. Just diluting with fresh medium is usually enough to replenish nutrients. And for spinner cultures, adding a surfactant like chloronic F68 is often recommended to protect the cells from shear stress caused by the impeller.
Good tip. Okay, so we can keep them growing. But what about saving them for later? Or as a backup, cryopreservation.
Absolutely essential technique. Cryopreservation, freezing cells for long-term storage. Why do it? Loads of reasons.
Like
Maintaining genetic stability, especially for continuous lines that can change over many passages. Saving finite lines before they set S. Having a backup in case of contamination, a huge one. Or equipment failure. Like an incubator dying, it protects your valuable resource.
A cellular insurance policy.
Exactly. You typically create a master seed stock from early passage cells, freeze that down carefully, and then thaw one vial from that to create working stocks for your experiments. Keeps the seed stock pristine.
Smart. How does freezing actually work without killing the cells? Ice crystals sound bad.
Ice crystals are bad. That's the main challenge. The standard method involves freezing the cells slowly in their complete growth medium, but with an added cryoprotective agent. Most commonly, DMSO dimethyl sulfoxide or sometimes glycerol. These chemicals lower the freezing point and reduce the formation of damaging intracellular ice crystals as the temperature drops.
And you freeze them slowly.
Yes, that's critical. A controlled, slow cooling rate, about 1 degree Celsius per minute, allows water to move out of the cells gradually before it freezes. You can use special programmable freezers or simpler devices like a Mr. Frosty container that uses isopropyl alcohol to achieve that slow rate in a standard NSF 80 degrees C freezer.
Okay, slow freeze. Any other key points for freezing?
Freeze cells when they're healthy and dense, ideally over 90% viable and at a low passage number. Use sterile cryovials designed for ultra-low temperatures. Store them ultimately below NaNSF 70 degrees C, but liquid nitrogen storage around nitrogen 196 degrees C is the gold standard for long-term stability. Store them in the vapor phase above the liquid nitrogen. If possible, it's safer than submerged vials.
Safer? Why?
Occasionally, tiny amounts of liquid nitrogen can seep into improperly sealed vials. When you warm those up quickly, the nitrogen expands rapidly and the vial can explode. Rare, but dangerous. Always wear proper PPE face shield, insulated gloves when handling vials from liquid nitrogen.
Good safety reminder. Okay, so they're frozen. Now bringing them back thawing, that sounds delicate too.
It is. Thawing is actually quite stressful for the cells, maybe even more than freezing. The key here is speed.
Fast thaw, slow freeze.
Exactly. You want to thaw the vial as quickly as possible. Usually take it straight from the nitrogen or freezer and plunge it into a 37 degrees C water bath. Swirl it gently until just a tiny speck of ice is left, typically takes less than a minute.
Why so fast?
To minimize the time cells spend exposed to the concentrated cryoprotectant as the ice melts, and to avoid the formation of large ice crystals during rewarming.
Okay, rapid thaw. Then what? Get rid of the DMSO.
Yes, pretty quickly. DMSO is toxic at room temperature. Wipe the vial with ethanol, then carefully transfer the thawed cell suspension, usually dropwise, into a tube containing several milliliters of pre-warmed complete growth medium. This dilutes the DMSO immediately.
Dilute, then centrifuge.
Right. Spin the cells down gently, pour off the supernatant containing the diluted freezing medium, and resuspend the cell pellet in fresh, warm, complete medium. Then plate them out into a new flask. Often good to plate them a bit denser than usual for the first day or two, helps them recover. And again, strict aseptic technique throughout.
Fast thaw, gentle dilution, careful handling. Got it. Now, the source mentioned a couple of other basic techniques, counting cells and checking viability.
Yeah, fundamental skills. Counting cells is usually done with a hemocytometer. It's a special microscope slide with a grid of known dimensions etched onto it.
Like a little counting chamber.
Exactly. You load a small sample of your cell suspension, and then under the microscope, you count the number of cells within specific squares of the grid. Knowing the volume of liquid over that grid area lets you calculate the cell concentration cells per milliliter. Crucial for accurate seeding.
Makes sense. And viability. How do you know how many are alive?
The most common way is trypan blue exclusion. Trypan blue is a dye. Live cells have intact membranes and can bump the dye out so they stay clear. Dead or dying cells have leaky membranes, so the blue dye gets in and stains them blue.
So you mixed your cells with trypan blue.
Right. Then you load the hemocytometer and count both the total number of cells, clear plus blue, and the number of blue dead cells. The percentage of clear cells gives you your percent viability. Aiming for 90% is usually good.
Simple but effective. Okay, one last thing the source touched on, troubleshooting. When things go wrong.
Yeah, it happens. Common issues are cells growing slowly or obvious contamination.
If they're growing slow.
First things to check, are you using the right medium? Is it fresh? Has the specific lot maybe expired or been reported to have issues? Did you warm it properly? Are your incubator conditions, temp, CO2, correct? Did you passage them at the right density or let them overgrow last time? Are they getting old? High passage numbers.
Basic checks first.
Always. In contamination, if you see it, usually best to discard the culture safely and start again from a clean frozen stock. If you suspect sneaky mycoplasma, you'll need to test for it specifically. Good record keeping really helps pinpoint problems too.
Definitely seems like attention to detail is everything.
Yeah.
Okay, wow, that was a comprehensive dive into the world of cell culture. Let's try to boil it down. What are the absolute key takeaways for our listeners?
I'd say 1. Cell culture is about growing cells outside the body in a very controlled artificial environment. 2. Success hinges massively on strict aseptic technique to prevent contamination. 3. You have to provide the right conditions, nutrients, gases, pH, temperature tailored to your specific cells. 4, understanding growth phases and proper subculturing is vital for maintenance. And 5, cryopreservation is your insurance policy for long-term work.
And for everyone listening from Aladdin Creations, especially you medical and lab science students, hopefully you see now this isn't just some niche lab trick. It's fundamental. Understanding this opens doors to research, diagnostics, drug development, tissue engineering. So many fields rely on these basic principles.
Absolutely. Mastering these basics gives you a powerful tool set. So if anything we talked about sparked your interest, maybe a specific technique or a type of cell, definitely dig deeper. Check out the resources. Look into protocols relevant to your field. It's a huge and fascinating area.
For sure. And maybe a final thought to leave you with. This ability to cultivate and study living cells in a dish. It's revolutionized biology and medicine, and it's still evolving. The knowledge you gain here isn't just academic, it's equipping you to potentially be part of the next wave of discoveries tackling major health challenges. Pretty exciting stuff.